There is a patient, intelligent, health-conscious, the kind of person who reads studies, tracks biomarkers, listens to every major longevity podcast, and considers themselves far more sophisticated about their body than the average person walking into a GP's office and accepting whatever they're handed. This patient was offered a statin by their cardiologist, studied in 170,000 people, backed by a 124,000-person meta-analysis that specifically examined and found no evidence for the side effects she feared most. A drug with one of the most comprehensive human safety and efficacy profiles of any pharmaceutical ever developed. She refused it. Instead, she was injecting BPC-157, a compound with human data from a study of 14 people, purchased from a gray market online vendor, based on the recommendation of someone who was financially profiting from its sale. This is not a story about stupidity. This is a story about a systematic failure of risk assessment, a failure that is happening at enormous scale in the biohacking and performance optimization community right now, in 2026. And that involves a category of intervention that could have serious, and as yet, entirely unquantifiable consequences for the people taking it. The peptide boom is real. The market for injectable peptides, compounds that range from genuinely transformative, evidence-backed pharmaceuticals to research chemicals with no meaningful human safety data, has exploded over the last 3 years. In February 2026, HHS reversed the FDA's 2023 restrictions on a range of compounded peptides, restoring 14 previously restricted compounds to legal compounding status. The regulatory environment has changed. The cultural enthusiasm has been building for years. And the content ecosystem, podcasts, Instagram accounts, YouTube channels, longevity clinics, functional medicine practitioners, has amplified the perceived promise of these compounds to an audience that is hungry for the next edge. I want to give you the most honest, most evidence-grounded account of the peptide landscape that exists anywhere in the content space. Not a dismissal of the entire category. Some of these compounds have genuinely compelling evidence and transformative potential. Not an endorsement of the entire category. Some of these compounds have theoretical risk profiles that nobody in the space is discussing with the seriousness they deserve. What I want to give you is a framework for thinking about evidence, risk, and the gap between what these compounds are being claimed to do and what the research can actually support. Let me start with the distinction that everything else depends on. There are peptides with robust, large-scale, peer-reviewed human clinical trial evidence. And there are peptides with primarily or exclusively animal model data, theoretical mechanistic rationale, and the kind of small-scale, often clinically uncontrolled case series that function in the alternative medicine space as evidence, but that would not pass any reasonable scientific standard for confidence in safety or efficacy in human beings. These are not the same thing. But in the way they are marketed, discussed, and presented to the consuming public, they are frequently treated as if they exist on the same evidence continuum. They do not. The established peptides, the ones with robust human evidence, include the GLP-1 receptor agonists, semaglutide, tirzepatide, liraglutide. These are peptide-based drugs that have been studied in tens of thousands of people in Phase 3 randomized controlled trials. Semaglutide alone has been evaluated in the SUSTAIN program for diabetes, the STEP program for obesity, and the SELECT cardiovascular outcomes trial involving more than 17,000 participants. The evidence profile is not merely positive, it is transformative. Average weight loss of 15 to 22% of body weight, meaningful cardiovascular risk reduction, emerging evidence for neurological and liver benefits. These compounds have earned their status through the most rigorous evidentiary process in medicine. Ipamorelin, a synthetic analog of growth hormone-releasing hormone, FDA approved since 2010 for visceral fat reduction in HIV-associated lipodystrophy, represents a different category of established peptide, one with meaningful human data for a specific clinical indication with defined safety monitoring parameters, and with an expanding off-label investigation that is at least grounded in clinical supervision and ongoing data collection. These are the peptides you hear about in the biohacking space that actually have the evidence to back what is claimed for them. The GLP-1s and Ipamorelin exist in a genuinely different evidential universe from what I am about to describe. Now let me take you into the underground. BPC 157, Body Protection Compound 157, a 15 amino acid peptide derived from a protein found in human gastric juice. In the biohacking and performance optimization world, it is described as capable of healing tendons, repairing ligaments, reversing gut damage, protecting the brain, improving mood, and accelerating recovery from essentially any tissue injury. The enthusiasm is extraordinary. The injection protocols shared on forums and in practice are specific and confident. The testimonials are effusive. The human evidence, as of the most comprehensive systematic review available in 2025, there is one human study of BPC 157 with an outcome measure. 12 patients received intra-articular injections for unspecified knee pain. 7 reported subjective improvement over 6 to 12 months. That is the sum total of the controlled human evidence base for the compound that thousands of people are currently injecting into themselves weekly. The preclinical evidence, the animal model data, is genuinely interesting. Studies in rats and other animal models show real, measurable effects on tendon healing, ligament repair, muscle recovery, gastrointestinal protection, and neurological function. The compound does something in animal models. Multiple independent research groups have replicated findings. The mechanistic basis is plausible. The preclinical literature is not fabricated. But preclinical evidence is not human evidence. This is not a technicality or a regulatory formality. It is the central fact of modern biomedical research. The history of medicine is littered with compounds that produced extraordinary results in animal models and either failed to produce human benefit in controlled trials or produced human harm that the animal models did not predict. The translation from rat to human is not assumed. It has to be demonstrated. And for BPC 157, it has not been. More importantly, the animal literature that BPC 157 supporters cite for its benefits also contains findings that they rarely cite. The compound promotes angiogenesis, the formation of new blood vessels. In the context of tissue healing, this is understood as beneficial. New blood vessels feed the healing tissue. But angiogenesis is also one of the core mechanisms through which solid tumors grow, spread, and resist therapeutic intervention. A compound that promotes angiogenesis in a healthy young athlete who is recovering from a tendon injury is doing the same thing to any undetected pre-malignant cells that may exist in that person's body. The angiogenic signaling does not read intentions. It does not selectively promote healing in the intended tissue while leaving cancer biology untouched. This is not a claim that BPC 157 causes cancer. We do not have the human data to make that claim in either direction. That is precisely the problem. We do not know. And we do not know in a context where the theoretical risk is not trivial. It is grounded in the compound's documented primary mechanism of action. The FDA classified BPC 157 as a substance prohibited from human compounding. The World Anti-Doping Agency banned it in 2022 under the SO category, the category for substances not yet approved by any regulatory authority for human use. These are not the actions of regulatory bodies that are unaware of the compound's potential. They are the actions of regulatory bodies that have reviewed the available evidence and concluded that the human safety profile is insufficiently established to permit clinical use outside of controlled trial settings. In February 2026, the regulatory landscape shifted. HHS Secretary Kennedy announced the reversal of the FDA's 2023 restriction on 14 compounded peptides. BPC 157's status in the evolving regulatory environment is actively contested. But regulatory permissibility is not the same as evidence of safety or efficacy. A compound that is legal to compound is not thereby proven safe or effective. It is legal to compound. The evidence question is separate. TB500, thymosin beta-4, sold under the TB500 brand name, is another compound widely used in biohacking circles for recovery and injury healing. The preclinical data on TB500 was evaluated for clinical development and produced a finding that should give any thoughtful person pause. In animal experiments, TB500 was found to accelerate the growth of dormant tumors and disrupt normal immune responses. This is not a theoretical concern extrapolated from mechanism. This is an empirically observed finding in the actual preclinical research. The FDA classified TB500 as a substance with specific safety concerns and prohibited its compounding for human use. This is a compound that is currently being purchased from gray market vendors and injected by athletes and biohackers who may not be aware that the animal research on the compound they are using to heal their rotator cuff found that it also woke up dormant tumors. The growth hormone-releasing peptides, CJC1295 and Ipamorelin, frequently sold and prescribed as a combination stack, represent a different risk profile, but one with an analogous problem of evidence. These compounds work by stimulating the pituitary gland to secrete growth hormone. Their claimed benefits, increased muscle mass, improved fat metabolism, enhanced recovery, anti-aging effects, follow logically from the elevation of circulating growth hormone and IGF-1 that they produce. The risk concern is equally logical. Growth hormone and IGF-1 promote cell growth. They promote it broadly, in the cells that need to grow to repair muscle, and in the cells that, in a person with undetected malignancy or pre-malignancy, should not be growing. The IGF-1 axis is one of the most studied mechanisms in cancer biology. Elevated IGF-1 is associated with increased risk of colorectal, breast, and prostate cancers in multiple large epidemiological studies. The link is not proof that raising IGF-1 through peptide administration causes cancer. It is the reason why broad stimulation of the growth hormone axis in healthy people who are not being monitored for cancer biology should not be treated as without risk. Eric Topol, the cardiologist and founder of the Scripps Research Translational Institute and one of the most respected voices in evidence-based medicine in the world, published a comprehensive analysis of the peptide boom in which he made the key observation directly. There is no evidence from randomized trials in humans that any of the underground peptides, BPC 157, CJC 1295, Ipamorelin, TB 500, and others, provide the benefits that are advocated for them. Not insufficient evidence. No evidence. And across all of them, the risks, wrong dose, unknown purity, potential contaminants from unregulated manufacturing, and the theoretical or documented biological risks I have described, are entirely unquantified. The dosing problem deserves more attention than it gets. When a physician prescribes semaglutide, the dose is based on the results of pharmacokinetic studies conducted in hundreds of people, refined through Phase 2 dose-ranging trials, and validated in Phase 3 safety and efficacy trials. The dose being prescribed is a dose that has been demonstrated in human beings to produce the intended effect at an acceptable risk profile. When someone injects BPC 157 at a dose described in a forum post based on another person's experience with a compound purchased from a vendor whose manufacturing standards are entirely opaque, there is no pharmacokinetic data, no dose-response curve established in humans, no quality control verification, and no meaningful way to know whether the dose being injected is the dose that was actually measured and loaded. Unregulated manufacturing is not a minor concern. Peptides degrade. They require specific storage conditions. They are subject to contamination during synthesis. The peptide sold by a gray market research chemical vendor at a price far below clinical-grade compounding costs is not the same as a pharmaceutical-grade compound manufactured under FDA oversight. The gap between what is on the label and what is in the syringe may be a problem of potency, of purity, or of contamination. And any of these problems is fully invisible to the person injecting it. I want to give you the decision framework that I think should guide any serious, intelligent, health-conscious person navigating this space. The first question to ask about any peptide you are considering is, what is the human evidence? Not rat studies, not mechanistic plausibility, not forum testimonials, not the confidence of whoever is selling or prescribing it. Human trials, conducted by researchers without financial interest in the outcome, with a sample size large enough to detect meaningful effects and a design rigorous enough to control for placebo response. If the answer to that question is, there are no human trials, or there is one small uncontrolled case series conducted by the people selling the compound. That is not a foundation for injecting an unknown quantity of an unregulated substance into your body. That is a foundation for waiting until the human evidence exists. The second question is, what are the theoretical risks, and are they trivial? For the GLP-1s, the theoretical risks are known, quantified, and manageable under clinical supervision. For BPC 157 and CJC 1295, and Ipamorelin and TB 500, the theoretical risks include cancer biology that is not trivial, not quantified, and not manageable because the trials that would quantify and allow management of those risks have not been conducted. The third question is, who profits from your belief that the evidence is stronger than it is? The people selling and prescribing underground peptides profit from the perception that the animal data is sufficient, that the regulatory restrictions are overcautious, and that the thousands of anecdotal success reports constitute evidence of safety and efficacy. They are financially motivated to present the evidence in the best possible light. That does not mean the compounds are useless. It means that you should not trust evidence that comes exclusively from people who benefit from your positive assessment of it. The peptide space is not binary. It is not all established medicine versus all fraudulent quackery. The GLP-1s are extraordinary and are changing medicine in ways that are genuinely historic. Tesamorelin has real evidence for specific indications. There are compounds currently in formal clinical trials, MOTSC, retatrutide, and others, where human data is actively accumulating and where the evidence picture will look substantially different in 3 to 5 years. The practical position for a serious person in 2026 is this: use the established peptides where they apply to your situation, under appropriate medical supervision, with the monitoring that the evidence says is appropriate. Watch the clinical trial pipeline for the compounds that are currently investigational, and for the compounds that are being injected by thousands of people based on animal data, form consensus, and the confident recommendations of people who profit from selling them. Ask the question that the patient who refused a statin did not ask about the compound she chose instead. How many humans has this been tested in? And who paid for the testing? Those two questions will tell you almost everything you need to know.